This is a production of Cornell University.
Thank you very much, Jeff, for that generous intro, and I hope I can at least somewhat appease everyone since they're here, okay? So one of the greatest challenges that we have in the 21st century, as you all know, in plants is trying to connect genotype to phenotype. But also, I think one other grand challenge we have is trying to feed more people highly nutritious food. And where my research lies is in crop nutritional quality, high-throughput phenotyping, and analyzing quantitative complex trait variation in crops. So the large thrust of my presentation will focus on these two parts right here, and there'll be parts in terms of like GS and GWAS as well. So when we think of the world's population now, we're at 7 billion people. But for the ones that are in the less developed country, we're at 5.8 billion. And when you think about the people who are not getting enough calories in the foods that they're eating, or they aren't eating foods, it's over 1 billion people. But where I draw my passion and where I kind of drive my research right now, and what is the thing that really gets me up out of bed every day early in the morning, is trying to work on this question of 2 billion malnourished people in the world. And when we think about this, we can think of things that are extreme. So there are these populations in the world, be it in South America, Asia, or sub-Saharan Africa, that are highly deficient in things such as vitamin A, iron, zinc, and iodine. And then things that are just like chronic but very much targeted in certain subpopulations would be such as vitamin E and B vitamins. So of the traits that I focus on in my lab are generally provitamin A, iron, zinc, vitamin E, and B vitamins. And so for the past almost 8 years now, my colleagues and my group have been trying to figure out ways to improve variation for these traits in maize. But before we go there, I would like for you to think about what could be a possible panacea for solving this grand challenge that we have in trying to improve the world's nutritional value of the food crops that they eat. We can think of giving pre-made vitamins out. We can think of having the food processed and we're artificially adding vitamins to the food. We can also think of increasing the amount of fruits and vegetables that people are eating, or perhaps fish and eggs. So all of these are part of the puzzle, and the one I would like to focus on now, biofortification, and that's where we're trying to use variation in plant populations, being genes, to try to improve the quantity of these different nutritional compounds. So it is certainly possible one could take a transgenic approach to this. And now you can think about even in the future after we find the causative variants, such as a CRISPR approach could also be used. But in the case of maize, which I will now focus on, we've been able to use the populations that already are present, say, in the tropical and temperate parts of the world, and harnessing that variation in plant breeding populations without having to take a transgenic approach. But when we think about maize, we all love maize. It drives our economy here in the US. But there are some countries, such as Zambia, here in orange, where over 50% of their calories are coming from maize. And why this is a possible issue is, when we think about maize, it's a great source of carbohydrates, those calories that I was talking of earlier. But what it lacks is a lot of the nutritional components that these at-risk populations very much need, such as vitamin A, vitamin E, B vitamins, iron, and zinc. The great thing about maize is it has tremendous natural variation for these important phenotypes for this world's population. So we can think about harnessing it in a biofortification approach. So what that first requires is trying to find the genes and, in a sense, at the end, the causative variants that are controlling this variation in these plant populations. So one of the traits that we've heavily focused on in my group is provitamin A. And after it's consumed, say, by humans, it is then converted into what we now know as vitamin A. But let's, at first, look at the impact of vitamin A in the world. When you think of these at-risk populations that are not getting enough vitamin A in the foods that they eat, there are 7 million pregnant women, 127 million preschool-age children are vitamin A deficient. And if you look at this green area right here, in such as like Zambia, where, as I was saying earlier, half of their calories are coming from maize. And there are at least like 20% of the population is severe for being deficient in vitamin A. Vitamin A deficiency causes 600,000 early childhood deaths each year and blindness in 500,000 children each year. So the thing that drives us when you are a plant breeder, you have the opportunity to create something that can be of value to humanity. And this is, in a sense, where my passion lies, because all of this is preventable, and this is something we can tackle as plant breeders. But with trying to ask any question when it comes to plant breeding, you have to ask the question, what is the composition for a particular compound in the grain? In this case, we're looking at carotenoids. And if you look here, the highest proportion are zeaxanthin and lutein. Those are obviously very important for the health of the eye, but they have no provitamin A activity at all. So of the compounds here, there are basically 3 which have provitamin A activity, but my favorite, and the one that has highest provitamin A activity, our friend beta-carotene right here. But as you see here, it's only 6% of the total of the carotenoids found in grain of maize. Now let's think about it in terms of the continuous variation in a population for this compound. So on average, in a population of temperate and tropical lines that is capturing a lot of the variation in the public breeding pool, on average, 1.7. So to put that into context, Harvest Plus has a target of 10-fold that. So on average, we need to increase beta-carotene by 10-fold. As you see here, though, at the tail, it's only 7.5, okay? So what you should then be asking yourself as plant breeders is there superior alleles that we can recombine in these, like, say, tropical maize germplasm populations that we can increase provitamin A to or even greater than this target that's been set by Harvest Plus. So that's a challenge that we have here. Now the other trait we've been working on in my lab is vitamin E. So there's something that needs to be clearly defined here. There aren't really extreme deficient populations in the world for vitamin E. There are populations in various countries that are not getting enough vitamin E in the foods that they eat. So even in the US here, there are 26 or 40% of people that had plasma tocopherol at a concentration that was actually lead to an increase of cardiovascular disease. That's 26 to 41% in the US. And when you think about it now in terms across the world, there hasn't been a lot of work on vitamin E as there's been on vitamin A. So we don't really have a good handle on what are the at-risk populations across the world. But for what has been reported, we see that there are some countries such as Brazil, and who knows what this country is? Tunisia. Very good, Elodie. Very, very good. Obviously, the French are very well trained on their geography. So here we can ask the same question. And in this case, alpha-tocopherol has the highest vitamin E activity in grain of maize. But as we see here, only 11% of all of the tocochromanols, i.e. vitamin E compounds, are comprising what you would actually find in grain of maize. But then when we look at the continuous variation, as we did with the provitamin A, here we have an average of 8.19. So if we can do some calculations based on eating 400 grams per day on what someone would require, on average, you're talking about breeding like 4 to maybe 5-fold higher on average in a population. So this is something that it actually seems to be more tractable, right? But there's a catch that I will show in a few upcoming slides. So now we have the challenge here. We want to find what these causative genes are, and we also want to find what are the superior alleles controlling this variation in these populations. And everyone's favorite hammer, the maize NAM population. Here it has been genotyped with HapMap version 1 and version 2. And we've had 2 outgrowths of this population. And if we can include all the checks and controls, we are pushing close to 20,000 HPLC runs on this seed, which, as you would imagine, required chaining 4 postdocs to 4 HPLCs for 2 years. Of course, they weren't chained, but that's just to kind of give you an idea here. But so the positive here, if there is one, we can actually phenotype carotenoids and tocochromanols at the same time from the same grain sample. And one other thing of value is here, so we have the HapMap version 1 and 2, the HPLC data, and we've done RNA-seq on 6 time points on grain after it has been pollinated. So you can think about over these 6 time points of the 25 founder lines of the population here. And so what that then gives us the chance to really do here is ask the question, in a sense, what genes are turned on and off over the phase of kernels. But also it allows us to ask the question for the QTLs that we find, which ones are actually eQTL, and when these eQTL are actually turned on. And so obviously I can show hundreds of these plots, but I will just be showing 2. So here, what we in a sense are finding is for our favorite provitamin A compound, beta-carotene, for this one that has the highest provitamin A activity, there are only 7 QTL that are controlling that phenotype. So as you should be thinking as breeders, this is something then that is very tractable, because when you think something as complex in yield, there could be hundreds to perhaps thousands of genes controlling that variation. But for this, in terms of like the composition trait in the grain itself, we are only finding 7 QTLs in this population. But I would also like to offer that there are most likely rare variants out there that haven't been captured in this population that could be contributing to variation as well, when you think about it as a crop. So of these QTL, we found 5 hits that are in very well-defined candidate genes from the carotenoid pathway. So that gives us a pretty good feeling. And these 5 QTLs, which had what we will refer to as a priori genes, had very large effects as well, whereas these 2 that are unresolved had effect sizes in terms of across the population that were actually very small. So in a sense, we're able to define and determine what these causative genes actually are for the key QTL controlling this trait in this population. And this is where the RNA-seq data can become very valuable, because then we can begin putting all the QTLs on a time axis. And so what we are seeing here, these are the effect estimates for the QTL themselves, and this is the log transform FPKM value for the gene. Okay, so you can kind of view it like copies for the gene in each of these founder lines here. But what we're seeing here for this one gene, this CRIT RB1, which is, as you see here, is converting beta-carotene to beta-cryptoxanthin, which has only half of the provitamin A activity as this compound here. And this is very much an eQTL, and it's constant over the whole, if you will, phase of the kernel. Okay, so here it's turned on at 12 days after pollination. Here, 20 days after pollination. So the thing here that's very valuable is, so we're testing the effect sizes or the effects themselves of the QTL with the gene activity. Okay, so in a sense, we're able to then say that this is in fact an eQTL. And since 5 of the 7 QTL were controlling variation and from a pathway, we now have the opportunity to really design a pathway breeding approach to improving provitamin A in maize. And here, in this case, we had these 2 genes right here, which you can kind of view them as you want them turned on very highly because they are generating the precursors. Okay? And this is a PSY1 gene right here. This is the first enzyme going into the carotenoid pathway, and you want this gene to be turned on very high as well, because you want to take all of the flux from here and have it going down into the carotenoid pathway. And then if we have an allele of this epsilon gene right here that is weak, then the activity of this gene will be low, and then the flux will then shift over to this branch point over here. And why that is important, because this is a side with beta-carotene, which has the highest provitamin A activity, right? And so we have to have a break in our pathway too. So this is where the CRIT RB1 comes into play. If we have a weak allele of this gene here, that means less beta-carotene will be converted to this compound, which has only half of the provitamin A activity. So now when we think about breeding for high provitamin A content, it becomes very much a combinatorial breeding approach when you're just trying to recombine the most optimal variation at each of these genes to get the highest quantity of provitamin A in maize grain. And in, I would say in the past 5 to 6 years, these 2 genes here have been used to release high provitamin A hybrids to the country of Zambia. And now, having worked with very much closely with Harvest Plus with my colleagues, now there are over 10,000 farmers in Zambia that are growing this orange high provitamin maize. So I think one thing that's obviously very valuable when you're being in a plant breeding department is obviously you want to do cutting edge science, but we have to reduce what we are doing to practice to have an impact on the people that we are trying to help. And I think one of the key points here is that if we just created something that's kind of like a foreign concept of high provitamin A, orange maize, and brought it to a country like Zambia, they probably wouldn't eat it because, say, for the past 100 years, they have been eating grain that is white from maize and in a sense has no provitamin A at all. Okay? So there's like a strong cultural preference to eat the grain that is white. But because of outreach and education from groups like Harvest Plus, they actually go into the towns and they educate the end user that eating this maize will save the eyesight of your children and perhaps also their lives. Because if you don't bring the public in from the very beginning, we cannot expect that they will use something that we have created over the past 10 years. So now Christine in my lab has been carrying out some work where we have been trying to ask the question, okay, we've been breeding for high provitamin A. What happens when this pathway, which we have now characterized very well, is being perturbed by heat and drought? And that's some of the things that she's been carrying out now. And as I'm pretty sure the seeds have been planted, the plants are germinating and they are growing, and we have everything in place that we will be receiving shipped grain coming in this coming year, and then we will run HPLC on it. But that, the thing we have to ask ourselves with like climate change as well, where we've been testing all these high provitamin A lines under well-irrigated, happy places, right? Now let's put them out in places where they have to undergo heat and drought, which is very much going to be a reality that the plants and the people will face going forward. So now to vitamin E. And what I showed earlier was a very tractable trait in the sense we were able to find what the causative gene was for those 5 QTL. Now for alpha-tocopherol, which has the highest vitamin E activity, this is a case where 13 QTL are controlled its phenotype, and when we look to see what the candidate genes are, we only can figure out 2 of them. One thing I would also like to point out is this VTE4 gene, it's able to capture half of the phenotypic variation that we see in this population, whereas this one candidate gene here and all of the ones that we can't characterize, their percent variation accounting for the phenotype is actually 2% or less. So we have now really tried hardly to like tease apart these QTLs. The thing about plants, they like to keep things like this very tightly to the vest, okay? So these are the things that are very challenging to tease apart. So of these, we have, I would say, 5 very good candidates that we have been able to tease apart, and it seems like transport is actually fairly key in the cell and for this pathway. So this is in contrast to the CRIT RB1 approach, whereas here, 12 days after pollination, we don't really see any trend at all. In a sense, the activity that the gene being turned on isn't tracking with the effects of the QTL at all. Only 20 days after pollination do you really see that enzyme begin to convert gamma-tocopherol to the compound that has the highest vitamin E activity. So this, as I was saying earlier, we can then begin to have an idea of when these QTL are turned on. So then we can then have hypotheses that can be tested where then perhaps we can find a QTL that is turned on 12 days after pollination. And then we can ask the question, is that going to lead to higher content in the grain itself? So those are things that we are asking now. And after now carrying this forward, our grant was just funded. So here we've been previously working on provitamin A, vitamin E. Now we're beginning to tackle B vitamins and zinc and iron. And this is by Di Wu in our lab. As you see here, we're beginning to analyze B1, B2, B3, and B6 in the grain of maize. And in the 281 panel, we're finding quite a bit of variation. So we think when we go to a panel of, say, over 2,000 lines, we'll be able to find greater variation. And these blue ones right here, these are the lines that we are employing in crosses now, because we're trying to ask the question, why is this an extreme phenotypic outlier? Is it a QTL that's, how does it actually work? So that's what we're trying to figure out now. And this is a GWAS that was run just this past few weeks in the maize Ames panel. And Di Wu, I'm happy to say you have some hits here that look very, very promising that we'll be able to follow up for the trait of iron. And why this trait is very important, iron deficiency impacts over 2 billion people worldwide. So this is like one of the most important traits that we could be breeding for in maize. And all of this information now that we are generating in provitamin A, carotenoids, vitamin E, tocochromanols, B vitamins, zinc and iron are actually now being able to feed into my own breeding program, because the third most important vegetable in the US isn't tomato, isn't potato, it's sweet corn. I know, I was surprised as well. And would you believe people eat over 20 pounds of it per year? I don't consider myself one of those people, but hey, there's some people that could be eating quite a bit of corn, okay? So the thing that we have been doing for the past 2 years is growing a very large panel out in the field. And since these traits, we have to phenotype on fresh kernels, right? So this is a little more challenging than what we had been doing in a sense in like field corn maize. We have to harvest it fresh, and then the ears themselves are in a sense kind of frozen out in the field, and then they're ground up frozen, and then we run HPLC on it. Thank you, Matt, for doing all this hard work. And all of this is actually beginning to turn into some real GWAS hits. So our friend right here on chromosome 5, which is that key enzyme that converts gamma-tocopherol to the compound that has the highest vitamin E activity, we hit that same key gene in this panel. But the question we have now, though, is the most favorable variants already in this panel, or do we have to begin crossing between germplasm pools? And that's the real challenge, and we hope not, but if we have to, then we have to. And we are also doing GS for this breeding program, and there were 100 crosses this past year. Thank you again, Matt. So here we are now doing GS, and this, if you will, this is like provitamin A, and this is the combo of these 3 compounds that have the highest provitamin A activity. And these 2 compounds here, if you recall, are the ones that are very critical for eye health. So this is just for one HPLC run, and so we think this will improve over time. So here for this trait, it's at 0.6, and here for these 2 traits, which have high provitamin A activity, we're close to 0.2. But part of the challenge with this is this panel has been genotyped with GBS, and as we've seen in our prior work, we need to closely tag the causative genes, if you will, in the panel, and there are some genes we are not tagging very well. And so, sorry, Matt, we're going to have to do some targeted PCR to actually pull those out possibly, or we can just do whole genomes and call it a day. I'll have you choose which one you want to do. So, so the one critical question that I had back in 2009, after we were generating all these HAP maps, were, what about the phenotype? And that's when I began working on field-based high-throughput phenotyping in my prior job on cotton. Because even though we think with GS we're going to have to do a lot less field phenotyping, we have to do it for the training set, and we have to do it repeatedly over time to update the training models as well. Okay? So in my prior work, we created this high-clearance, high-throughput phenotyping tractor here, and everything is run on GPS, so we can figure out where the actual trait is coming from in a plot, and our field map is on a GPS coordinates as well, and we're able to look at vegetation variation from canopy reflectance, canopy temperature, and canopy height. And a great thing with this tractor was we were able to do it over the whole growing season, and I didn't have to be out there like 2 p.m. in the afternoon when it was 120 degrees Fahrenheit. We just had someone driving a tractor through. Just so you know, our revamped tractor, I'll just call it now, has a cab with AC on it. So all this looks very draconian, if you will. Someone's having to drive this tractor through the field. Things have been revised and they're improved now. So actually, it's all on GPS and everything, so the person in the tractor cab doesn't even have to touch the wheel. He or she just sits in there and it just drives by itself, and they're in AC, and for all I know, they could be having some like film fest in there or something, you know, because you don't have to see where you're driving at that point. And so this is where you have a question that you're asking and the tool isn't created. So I had the question in terms of like, how much variation is there for, say, canopy temperature, and kind of quantify that over time, over the whole growing season, and can I find QTL that is actually controlling that variation at each of the key time points over the growth phase of cotton. And here we had a recombinant inbred line population, so this was very humbling when I went into cotton. There were no very large populations, so I had to use what was basically created for me at first. And then here we had the high-throughput phenotyping tractor, and then we had a cotton picker driving through to do traits such as like yield, fiber quality traits, and some plant phys traits. Is Tim Setter here? I was going to give a shout out to him for thank you for doing this plant assay here, but I'll highlight that result soon. So here we have our actual test here. So here we had dry rep 1, which is basically our drought. This one here is our control treated as wet, then dry rep 2 and wet rep 2 right here. And the great thing is all growing on drip, and it hardly ever rains in Arizona. Although I will say, if you do plan a drought test, it increases the probability of rain, I have seen, even in a place like Arizona. And I think one other key point is we had to characterize the variation for the soil as well, such as like clay and sand content, because whenever you're doing any drought testing, the soil will have more variation than your actual plant population for the phenotype that you are testing. So just to kind of give you an idea of where we are here. So here we were at planting right here, and up until first flower, everything was treated the same in terms of the drip. And at flowering is where we began imposing the drought treatment. And the traits that we were also doing were canopy temperature. This is a vegetation indice here, and canopy height. And this is early in the morning, 10 a.m., 1 p.m., and in the afternoon. So in a sense, you're trying to capture the variation over the whole growing season here, as we're doing this every week, but also capturing the variation at each time point within each of those weeks. And so the three growth phases that we were targeting were at flowering, and as post-flower, and as we have the growing fiber as well. And then, of course, yield and the fiber quality traits that were collected as well. And so this was just published this year, and one of the fascinating things with this work is, prior to this, there really wasn't a way to quantify a trait like canopy temperature in the field. It had to be all by hand. But traits like this can actually change with time very rapidly. So then you could think you would maybe have to have an army of people to do like 400 and some plots just for a trait as canopy temperature. But this case, the tractor could phenotype all of the plots in a half an hour. And here we have each of the years, so it's 2010, 2011, 2012, and this is the treatment wet, and this would be the dry drought treatment here. And if it's red, then we have a QTL in a sense that's increasing the canopy temperature. If it's blue, we have a QTL that's decreasing the canopy temperature then. So if Tim was here, he would be so excited that sialic acid actually hit one of our key canopy temperature QTL. And this is actually showing up pretty early on in the growth phase of the plant, and it's carrying forward towards harvest as well under the two treatments. And why that is important, because this is a plant hormone that is, in a sense, controlling transpiration rate. So since we also have a QTL that is going right here, it's a possible candidate gene. But as you're trying to find candidate genes, you should always have the caveat, you can always find a candidate gene, right? But this one, actually, you can tie in with the biology very well, and the actual compound itself maps to the same place as that canopy temperature QTL. And then we also wanted to ask the question, can we use this to also track with yield? And what we found out is the plants that take, in a sense, take a great risk. And I will say this, because these are the ones that plan to use a lot of water and continue to cool their canopy here. These are the plants that will be trying to conserve the water, and then they won't cool their canopy. So for the plants that are grown here, if you recall, everything is grown under drip in a hot, arid world, okay? Now, for the ones that are going to continue to transpire at a high rate, they're going to continue to fix carbon, and if they're going to continue to fix carbon, they are going to have higher yield. But if you are perhaps growing in a dry land part of the world, you would perhaps want a genotype that will conserve water early on, and then will, in a sense, use towards the end of the growing season, such as like grain fill or fill in this case for boll fiber. So what we also figured out was that canopy temperature at flowering was most predictive of yield at the end of the season. And the reason why that was, it's clear now to us that if there's high heat, what do you think is going to happen to that pollen? That pollen isn't going to be very viable, right? So that is kind of like what is happening here. So if you have a high heat event at flowering, it'll have a great impact on the pollen. The pollen will be inviable, and then you won't have any seed, i.e. cotton fiber, okay? So we now wanted to ask the question, after cotton's been defoliated, in a sense, you add a chemical and it drops all of the beautiful green leaves, you can then drive a combine through it here, right? So this is, in a sense, creating a cloud point map of all of the cotton plants in our field. And in a sense, what this is you take something that's like bright laser, you shine it at a plant, it comes back, time of flight, and then as it's returning, you calculate the time, and then you can recreate like a 3D map of the actual plant itself. So since this is the defoliated cotton, what I really wanted to do with this was actually try to count yield. And how do you do that? Count the cotton bolls. So here's a case where we have one plot now, and so since this cotton has been defoliated, you can see this right here. These are the cotton bolls right here. So this piece of equipment was kind of put on the tractor, and here the tractor is driving through the field, and we're recreating the geometry of the plants themselves. And then you can pull out one plant and then use a Euclidean clustering approach, and then you're using that to count the cotton bolls on the plant, which is, in a sense, yield. Because there are actually, cotton breeders that grow out hundreds of thousands of plots, and they don't want to combine all of those all at once, right? They just want to target just those like F2s or F4 plants out in the field. So this could be valuable for that. So now that was with cotton, and now with this maize. So we kind of had the question, how do we do plant pathology in a cornfield? It's all by eye. It's all with human beings. If I'm out in the field, I guarantee you, I probably won't be accurate like Tier if he's out in the field. So how do we move that subjectivity from plant phenotyping? And if you're wondering, this is the one that's the corn blight, okay? So can we find the actual blight out in the field? So this is where artificial intelligence actually comes into play, and then tying it with drones, because we should now kind of view drones as almost our colleagues and our co-workers. And so yes, it sounds a little frightening. So when you have this drone flying through a cornfield, can you have it so that it's taking a photo and then it's analyzing that photo, telling you if a certain pathogen is on that corn leaf, and then tell the actual grower where he or she should go to the field to either apply a chemical or actually, chop the plant down or something like that. So what we have here is an actual drone photo of a cornfield, and this would be a beautiful corn lesion right there, right? Though, the challenge is I can easily find this one. Can we train the AI to go through thousands and thousands of photos that have been taken over the course of the year to then find it? And I think one of the most painful things is training this AI, because you have to have a true and a fact plant pathologist that's telling yes, no, yes, no, yes, no. So training it, I think, is almost like the hardest part, and you may have to train on thousands of actual photos. And to kind of get an idea of how this works, probably the easiest way I can tell you is it's, in a sense, converting pixels into words, okay? Yes versus no. And so after the AI has been trained, we can then take a photo from the field that, thanks to Tier, that he took, and then we can apply the AI to that plant. And in this case, if it's black, that means probability of 0. If it's very bright white, probability of 1. So in this case, it's finding all of them on the leaves right here, and then it's finding even some right here on that leaf. So probably one of the real challenges is training this AI is when you're out in the field, you also have a lot of soil and other plant parts and fragments, if you will, covering the ground, and also weeds, okay? And then we can also ask the question, in the case where we don't see any, do you find any? And in this case, no. So how well are we doing? I think we're doing pretty well, but we can always be a lot better. But I'm kind of happy with that, but sorry. So now let's talk about robots. The 20th century was all about chemicals. The 21st is all going to be about robotics. And I think when we think about plant breeding, what we should be thinking about, and this is my colleague Josh, where you can then be creating these autonomous field robots that can do phenotyping, that are going through the fields, doing such traits like canopy temperature, canopy reflectance, and canopy geometry. And what that can begin, I'll actually really ask the question is, what QTLs are turned on over the entire growing season? And then when you think about calculating biomass yield, if the robot's driving through the field every day, you can then think about biomass yield every single day over the whole growing season. And then you can perhaps begin to really think apart, like tease apart G by E questions. And where I think this field is actually heading is, so we have the AI, and we have that thing that we call the breeder's eye. Let's use it to train low-cost field robots to actually begin to help select plants from the field. So in this concept here, we have this very low-cost robot with these thin carbon fiber legs. It can walk through the plots, right? So say we had a breeder that may walk through like 4,000 plots, and that breeder is, in a sense, choosing plants based on their height and plant pathology. If that breeder can then train the AI, you can then turn out these low-cost, cheap robots just to kind of like walk through the field, and then they can choose the plants for the breeder. And if the breeder isn't happy with the plants that were chosen, they can retrain the AI each time. So this is a concept now, but this is where I see the field in the future, and that's where I'm kind of trying to like drag the field of plant breeding on this side aspect of it. Now with that, there are a lot of colleagues to thank for this work. I won't be able to thank them all because I'm out of time because we had the computer problems at first. But with that, I'll thank you for your time.
Sure, Mike will take.
Yes, Lee.
So you mentioned the PSY is the key to control beta-carotene.
Yep.
Have you or anyone in any group in the Cornell identified the best PS1 allele?
Can you repeat that?
Yeah, so the question that Lee asked, did we find the most superior PSY1 gene? In a sense, I would say at this point in time, no. But there are at least 2 or 3 that we have seen, but I don't know if we found the one that is like the key one, and that's where we're hoping with like the Ames panel that we may actually find that, and if we weren't happy with it, then we'll just create our own in the future. So, yeah. Yes, Steer.
You don't have a beta-carotene pool by tamping down CRB1 expression.
Yes.
That, you know, if that limits the use of those is that that's also
No, it actually hasn't, because like the part of the seed where those genes are turned on, it hasn't had an impact on it at all. Yeah. Yes.
And given the length of carotenoids and ADA and all the work in the NAM population, is there already some existing resources you can use to predict the drought performance of some of the beta-carotene enhanced corn? Maybe already overlapping peaks or about the flux and how you perturb the ability to go into ADA?
Yeah, I kind of have seen some things that have been published, but I don't think they've, some of it's been in like greenhouses and growth chambers and things like that. So I think this has been kind of the key part, actually taking it out to the field and actually testing it. But yeah, there are some things that we think that we should see, and we'll see if we actually see them. Yeah. Yeah.
Have you found any difference in vitamin E and carotenoid levels after cooking? That only work on fresh. Most of them boil or grill their corn.
Yeah, that's phase 2. But yes.
I want to make
Yeah.
a comment on this.
Yeah.
There's a PNAS paper coming,
Mm-hmm.
and they showed increased vitamin E can greatly stable.
Oh.
beta-carotene.
Oh,
That's
cool.
beneficial.
Oh, good, good.
Repeat that.
So the thing about tocochromanols, it can enhance the longevity of some of the compounds in the seed, and that's what you're basically saying, right?
So what I'm saying is that beta-carotene stability is a big issue for post-harvest storage, and there's a PNAS paper that was published
Mhm.
showed that increased vitamin E can greatly stable beta-carotene
In maize or in other crops as well?
I think they showed in sorghum.
Oh, and sorghum, yeah, okay.
So I thought
Well,
that
good.
perfect fit.
Good, okay. Yeah, Susan.
What Lee just mentioned is very important to me because one of the many issues with the golden rice, which is the high beta-carotene rice, which doesn't make it naturally, it's always a transgenic, is that they can get the level, but it decreases over time in storage. And rice is traditionally dried down and stored over the course of the season, and by the time people eat it, it doesn't have the levels that they had anticipated. So if we can learn from maize
Yeah.
and work in maize that a high level of vitamin, or in sorghum, that a high level of vitamin E helps to stabilize it, that could be very, very significant for the ways in which we used to think so simplistically, even though we called it metabolic. It was a transformation effort that early on
Right.
involved 3 genes in rice, but it didn't understand the stabilization part of it.
Afterwards,
Very
yeah. Oh, yeah. And, we also see that same problem in the high, corn. But the thing is there is like an E and a G by E and a G component to it. And the G component, I think we can kind of get a handle of, but then there's like a large E component that I think is more complex.
Well, there's
Yeah.
also no G by G because in indica rice, it holds less well than in japonica. But
Oh,
it's in
okay.
the tropics where they grow indica that they need it. They don't need it in the temperate where they grow the japonica.
Yeah.
So there's clearly a G by G.
Yeah. Huh. That's
Anyway, it would be
intriguing.
good to do a little more comparison. I'd be interested.
Yeah, sure. Okay.
I'm going to ask if there's any question in Geneva. I think there is someone there.
Any questions from Geneva?
I don't see any.
No.
Anybody else? Any other questions for Mike? Okay, if not, let's thank him again.
Thanks.
This has been a production of Cornell University, on the web at cornell.edu.